They've already done it.
Okay, let's have a class today. Today we're not going to do any lab work. We're going to do a very roughly review for the lab midterm exam from the beginning to the last lab, and then we will spend in time talk about the last lab. Okay, so let's do some of the review first. So October 3rd will be lab midterm. Will be the material covered so far. So we want to talk about these things and do a review. Number one lab is microscope use. If you still remember, microscope use, we are using medium microscope, Staphylococcus aureus, and I believe it's a beer bluing Saccharomyces. We do the observation. So you need to understand in several things. Number one, what is total magnification? And the total magnification, you always remember, is the magnification of objective lens, multiple ocular lens. So I give you a very simple example. If objective lens is 100x and multiple by ocular lens is always 10x, the answer will be 1000x. This usually is the first question of the exam, okay? The second one, we want to talk about what is the resolution power. It is equals lambda divided by 2 and NA. Lambda is the light wavelength, and the NA is numerical aperture. Numerical aperture. And we did the calculation. The material what we are using here, 625 nanometer divided by 2 multiplied by 1.25 equals 206 nanometer. Okay. And always remember, this number should be the smaller the better. Because the smaller resolution power, which means strong resolution capability. Okay, that's something we mentioned. Now, number three, obviously, we talk about the oil immersion. And you need to know what's the function of the oil immersion is to remove air gap. Because during the light refraction, and the refraction index of oil is equals refraction index of glass, which is 1.5, that is larger than refraction index of air, that is 1.0. So when we put the oil there, which is connected with the glass to remove the air gap. Okay, that's oil immersion. And we have another thing we mentioned about what is parfocal. The parfocal is a very good characteristics of the light microscope, which means lens stay focused once focused. That's a very good characteristics. And the last one we talk about is working distance. And always remember, the working distance is decreased with increasing objective length. Of course, the magnification, okay? That is our lab one we talked about. Then we move on to the lab two. What we did for the lab two is simple step. So simple stain, we really started to touch bacteria. We're using the staining technology, and we also review the microscope use. So first thing first, why we can do a simple stain? Because the bacteria culture, after 35 degrees Celsius, 24 hours, it is negatively charged. And most of those dye, it is positively charged. So they could connect it with each other, tracking with each other, and showing you the color. The second question, what are those basic dye? Obviously we know crystal violet. That's one of them. Saffronin. And another one, methylene blue. And carbon fusion, all those, okay? And what the results will tell us for the simple stain, we will know the cell morphology only. And we also will know the arrangement. Now, what are the cell morphology we talk about here? Basically, we know it's a Koch cell. It could be rods. It could be vibro, etc. Okay, so that's a simple stain. We talk about all these things. Now we move on to a big lab, and I said it's very important, which is called the gram stain. And we practiced several times and also did the unknown, so you should be very familiar with that. And we always say we start from the major steps. Okay, the major steps, which is including the first step, is number one, crystal violet. Crystal violet, the function is a basic dye. The second step is we add a granite to make it stay strong, so we call it a mordant. And the number three is we using 95% alcohol. It is a decoloration process. And the last step is counterstain with saffron. Okay, and we said very important, everything is 60 seconds, except the alcohol decoloration 10 seconds, and the counterstain is also 60 seconds. We mentioned about that the time is flexible, but the procedure is important. And we also talk about the gram stain. You have to use a very young culture. Usually 24-48 hour fresh culture, because the mechanism is based on cell wall. Therefore, we had our first theory, which is we call it a cell wall theory. That is because gram-positive bacteria has a heavy peptidoglycan. So that can hold on crystal violet during the decoloration process. And the second theory we call it a lipid theory, and that is explained. The gram-negative bacteria have around like 10 to 15% of the lipids, and then that could be washed off during the decoloration process. Okay, so that's lipid theory. Now, because it is based on the cell wall theory, so we have to use a very young culture. Otherwise, if it's very old culture, what are we going to do? It will result gram variable stain or gram variable results, which means everything stains gram negative bacteria, looks like. Okay? And now we're also going to mention that gram stain, very important. We need to recording 3 items What are three items? Number one is gram positive or negative, which is gram reaction. Number two is cell morphology. And number three is cell arrangement. Now we did some of the lab work, so we have some of the examples. Number one, let's say we had an E. coli. E. coli is gram-negative, single short lots. And we did Staphylococcus aureus, and we know it is gram-positive, it is a coxile, it is a grape shape. Very interesting results. We also did the unknown to talk about that, the mixed culture. Okay, so that is something important for the gram stain. And so you're going to be a reviewer of these. We also could ask you the gram variable stain. There is a two reason. One of them is using very old culture. Another reason is 95% alcohol. Decoloration too long. Okay, that's going to be the reason. Then we move on to the lab 4. What we did for the lab 4 is endospore stain. So we're going to write here is lab 4. Lab 4 is endospore stain. Okay, what is the endospore stain we did? Number one, we need to know bacillus, species, and Clostridium. Those are the two major species has endospore. Second one, we need to know the endospore is a stress protection structure. It is not a productive, or we say reproductive function. So which means the mother cell does not mean the daughter cell will automatically have. Number three, we talk about the structure of the endospore, why it will be resistant in a stress environment, and the key thing is right in the middle. We have two items. Number one is calcium DPA, calcium diproconic acids, and the second one is small soluble acid protein, acid DNA protein actually. These are the two major items. We'll let them survive. Now we did the endospore stain. Remember how we do the endospore stain? What is the major difference compared to the other stain? The major step is heat steaming for 5 minutes, and with a dye called malachite green. This is the major steps, okay? Of course, you have to counterstain with saffron. Is the final counterstain. So, what will be the results of endospore looks like? If you still remember, we draw the picture before. What we find in our lab is we using the Bacillus subtilis. What we find is the endospore is kind of like a little bit greenish, and it's subterminal. And sometimes it's going to be a big bubble, okay? Both of these, so we say it's a subterminal. And we also talk about the location is genetic dependent. the genetic dependent, which means there could be terminal, there could be central, there also could be subterminal. And what we find in our lab, it looks like it is subterminal. Okay, that's what it looks like in those spores. Now, lots of the other places you see, it looks like a big smear, which is pink because of saffron in color. Okay. Last one, we also want to mention that in those spore stem, we need to use very old culture. Now, how old it is? At least 48 to 72 hours, because we want them to form in those spores. And the indifference between the gram stem need to use very young culture, because the CRE is based on the cell wall structure. Well, that's what we did for the endospore. And next one, lab 5. Okay, lab 5 we're going to draw here. What we did in lab 5 is transferring bacteria. Into medium. Okay, so how we did that, still remember? First thing first, we transfer them into a bacteria broth. Okay, this is the bacteria broth. Incubate 35 degrees Celsius, 24 hours. How do we know the bacteria is already growing there, is already survived there? We talk about 3 markers. What are the 3 markers? Do you still remember? Number 1, pellicles. Number 2 is cloudy area, so we call it a turbidity. Number 3, we often see is a precipitation. That's what we call it, a sediment. It's at the bottom, okay? That's the 3 markers for a bacteria to survive in a broth. And we also talk about a slants. What we did is a zigzag marker. The zigzag marker, which tells you bacteria grow, and this is a temporary storage. Now, last thing, it is very important. We talk about streak plating of bacteria to generate a single colony. And the single colony represents a pure culture. And do you still remember how you did that? It is very important. You should always know. How we did that? Very intensive at the beginning. Okay, that is course number 1. Then we have to flame the loop. To the second one, less intensive. Course number 2, and then less intensive for the third one. Course number 3, and the last one is finally more or less intensive. Okay, that's number 4. And then you will see a colony is forming there after incubation. And this colony indicates a pure culture. And don't forget, here you have to do flaming. Here you have to do flaming. And right here you also have to do flaming. Okay. Why we say it is important? Because this is serial dilution on solid medium. Okay. This is the whole blackboard, is the first part of our review. So I will take a picture, I'll put on the eCampus, then you guys can see it, and I'll remove some, and then become a second part, okay? So I'll take a picture. Every year there's a little bit difference. So I always take a picture for the new one. Yes, of course. Everything is for you. Okay. So this is the outline from lab 1 to lab 5. The video is also going to be there, but don't forget to check your lab notebook. Okay. We have 2 more labs to go. I will remove part of that, keep something there, because I'm going to draw everything otherwise. But every single lab today, I will be starting from the beginning to redraw it. Okay. Otherwise, you guys are spending like half an hour. Other lab we can only finish in half an hour. That's 10 minutes. That's not very fair. So we will be all make sure they all understand it. So I'll be cut down from right here. Okay. Okay. So next lab, next one. What we did? Lab 7 is unknown. Lab 6 is unknown. We already talked about that. Will be homework, so we don't do the review. Okay, it's not going to be in the exam to redo it again. Next one, we talk about. Remember, Lab 7 is the bacteria enumeration. Okay, what we did? We did the pore plating, and we also did the spread plating. And you know how to do it, but I have a quiz for you, okay? For the math, very simple math. Let's do a practice. This is my bacteria solution. 35 degrees Celsius, 24 hours. Let's say E. coli. Okay, I'm going to do a serial dilution. 3 tubes. Let's make it simple. Okay, but a little bit complicated. Okay. I have the first tube is 9.9 ml, 0.1% neutron broth. Second tube 9.0 ml, 0.1% neutron broth. The last tube is 9.9 ml, 0.1% neutron broth. So how I did the dilution? I'm adding 0.1 ml, go to the first one. We mix it very well. Then I'm gonna adding 1.0 ml, go to the second tube, mix very well. Then the last one, I'm adding 0.1, go there. Mix very well. And finally, I'm gonna add them into the agar plates. Agar plate number 1, number 2, and number 3. Okay, now you're gonna have to tell me the tube dilution. What is the first one? The tube dilution factor of the first one is 10 to the 2. So what's the final dilution factor of the plates? Is 10 to the 3. Remember, we are 0.1 ml adding, therefore, the final plates dilution is always 10 times more than the tube dilution. So how about this one? This is actually 10 to the 3, because in the middle I'm using 9.0 ml instead of 9.9, so the final plates dilution is 10 to the 4. How about the last one? I did it again. This is the tube dilution is 10 to the 5, and the final dilution factor of the plates is 10 to the 6. Okay? A little bit difficult compared to what you did in the lab. A little bit twisted. Okay, now let's see the results. I got let's say I get 400. Okay, the second tube, what I get is 40. The last one, I get only. Sorry, not that. The last one, I get only 2 colonies there. Let's see. Now, first of all, what is CFU means? That you have to know. Is that right? What's a CFU? Colony forming unit. Okay, that's colony forming unit, CFU. Now let's do the calculation. Which place you can use? Lots of people start to use a calculator to take the average. No, you're not going to use this one. It's too much. You're not going to use that one. This is too less. Remember we talked about what is the zone? The acceptable zone? 30 to 300. That is the acceptable zone. Okay, 30 to 300. So how many it is? This will be the one we're going to use it. So that is 40 multiplied by 10 to the fourth CFU per ml. And remember, when we do this calculation, we'll calculate the concentration. It is not the whole bacteria in the tube. This equals 4 multiplied by 10 to the fifth CFU per ml. And we have transferred to the log is approximately 5.5 log 10 CFU per ml. So this is the results calculation. Okay? That is an example for you if I want to do it. Now I will maybe make it easy, maybe there will be only two tubes then to see it. Okay? And also in the real exam, I may draw some dots there. Can you counting the colony there? You should can count CFU on plates. Okay, you should can do that. Okay, that's lab 7. We will do a little bit of the practice, the calculation. Okay, last one. I want to have enough space. Let me see how many space I can have. I want to have enough space. We want to talk about. Unfortunately, I have to remove this section. So I'm going to remove this section. I can rejoin it. I'll keep some of them. Okay. I'll take this part. Let me take this part, and then we talk about today's work. Okay. What we have? Lab number 8. This is the bacterial media. So what we have? First of all, what we have? Neutron agar. What is the neutron agar? Support media. Bacteria can grow around 10 to the 8 CFU per ml, except the fastidious bacteria. Number 2, what we have? Macanchi agar. Macanchi agar should be only gram-negative bacterial growth because the components is crystal violet, Q gram-positive bacteria. And remember, it can also differentiate 2 ways. What are those 2? Lactose fermentation positive and the lactose fermentation negative. Lactose fermentation positive, you're going to see the pink colony there. Some of you did a good job today, can see some of the pink. So that's pink colony. And this is colorless.
Okay. We cannot see the lactose positive and lactose negative. Yeah.
I realize that, that's what we moved. So lactose positive and the lactose negative. Okay? Now why it is pink? Because pH indicator, that's neutral red. Okay. Okay, so that's Macanchi agar. Now the next one, mannitol salt agar. Okay, mannitol salt agar, you have a mannitol. So we differentiate mannitol fermentation positive, mannitol fermentation negative. If mannitol fermentation positive, it is yellow color, and the mannitol fermentation negative, it is colorless. And then you have a salt. The salt will become halophile and also have a non-halophile. Okay. Now we talk about the blood agar. Blood agar, 5% sheep blood. For enrichment plus alpha, beta, gamma, hemolytic. Okay, that is what we talked about already. These bacteria, agar. What the results looks like. Sorry, I'll just tear these off. Okay, let's just take these off. I'll redraw to the next lab. Because I want to make sure you understand it very well. Okay, we have a table. We want to talk some of the results. Not all the results are perfect. Okay, we have neutron agar. Makanki agar. Mannitol salt agar. And the blood agar. And then we also have bacteria. What are the bacteria we have? We put it there. Okay, first of all, E. coli, lactobacillus, and I said this may be Staphylococcus aureus and the bacillus, and it may be Staphylococcus aureus, could be. And the last one is Citrobacter. Okay, what our results looks like. Okay, you need to use another line. Makanki agar, mannitol salt agar, and blood agar. Okay. It's a table. First of all, this is your neutron agar. Everybody look. It should all grow. Is that correct? Okay. So we put positive, 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 positive. No question about it. Let's look at your blood agar. First of all, everything should grow. Grow, 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 because it's an enrichment media. Can you see which bacteria is beta hemolytic? For us, it's bacillus. Is that right? But I tell you, for me, I think might be Staphylococcus aureus is contaminated with this bacillus. That's why I write essays there. Might be. But we have to respect your results. So our results is bacillus beta hemolytic. Because you see a transparent zone. Okay, others we just say gamma hemolytic. or none. Okay, that's both are fine. Okay, now the tricky thing. Makankiaga. I want to tell you something. Makankiaga is supposed to be bacillus and lactobacillus should not be grown because they are gram-positive bacteria. So ideally, these two should not grow. Sorry. In the middle two should not grow. The top and the bottom should grow. Ideally, because bacillus is gram-positive bacteria. Ideally, it shouldn't grow. And then what you see your results. Is that all pink? It's all pink colony. Lactose fermentation positive. And this is also lactose fermentation positive. Citrobacter is lactose fermentation positive. Now, in order to responsible for your results, you could put a question mark, positive, and lactose fermentation positive, bracket. You could record it like that, because that is our results. Nevertheless, is our results. Ideally, should not grow, but we see it. Okay? Mannitol salt agar. What looks like? This is one of our two students in the first bench they did. I think they did really good. Bacillus subtilis is yellow colony. Lactobacillus is pink. Citrobacter and the E. coli didn't grow. So what happened? In the Mannitol salt agar, this doesn't grow, this does not grow. Lactobacillus is grow, but the Mannitol fermentation negative, so it is pink. And the Bacillus subtilis, it is grow, Mannitol fermentation positive, it is yellow color. This is your results table. Okay, we said it one more time. We have 4 different bacteria, but I'll tell you, right in the middle, maybe some of cross-contamination, could be. So I write a bracket there. Neutral agar, there is no question, everything should grow because it's a supported media. Blood agar is an enriched media, everything should grow. What we are interested to see, which one is better hemolytic, is bacillus here. Okay, that's why it is better hemolytic. Now the tricky thing, we move to Makankiaga. It is not a surprise to us. E. coli and Citrobacter should be grown because both of these are gram-negative bacteria, and we see pink colony there. That's lactose fermentation positive. That is why we see pink colony. And this is pink colony. And ideally, lactobacillus shouldn't grow, so they didn't grow. Bacillus subtilis should not grow. However, we see it's lactose fermentation positive also, and they grow. So we add a bracket there, because that's the results what we see in the lab. Mannitol salt agar, E. coli and Citrobacter, they didn't grow. Possible reason is the salt. They're going to kill them because there's not halophiles. And then in the middle, you see bacillus is growing, but it's pink colony because Mannitol fermentation negative. And then you see the lactobacillus. Bacillus, you see it's positive, Mannitol fermentation positive. That's a yellow color. That's a results table. So we could ask you to fill. Now I have a question for you. Let's make it clear. If we are able to use Staphylococcus aureus, what will be the results looks like? If neutral agar should be grow, blood agar should be grow, and the beta hemolytic. Is the macaque agar will grow? No. Because this is gram-positive bacteria, so macaque agar shouldn't grow. Is the mannitol salt agar will grow? Yes, it will grow. And the mannitol fermentation positive, plus it should be yellow colony. So to be fair, to be not confuse you, the table you should record on your results table. In the exam, we basically going to test you E. coli, Citrobacter, and the Staphylococcus aureus. We're going to make your life easy to avoid the cross-contamination. Okay? That makes sense. And you can record your results in your notebook, which is respected to the lab results. Whatever we have, what it is what it is. However, I just want to tell you in the exam, to not confusing you, we will test E. coli, Citrobacter, and a theoretical Staphylococcus aureus. And these two, we will just talk about. Because in your lab stream, could be has a cross-contamination there. That's why some of you get different results. Here, you see the yellow colony there, is that right? And I see some of you have everything grow there in the mannitol salt agar. So it could be your contaminated, or it could be the strain contaminated. So for example, I gave you an example right here. This guy is grow everything. And this guy, you have a 2F there. So this mannitol salt agar, very surprisingly, E. coli and Citrobacter is growing. So that could be a contamination. So I don't know where it comes. Okay, so to be honest, we don't want to discuss about that. We will test you 1, 2, 3, only those three. And I think you should do very well. I will also ask my graduate students, Carly Long, to help you to do a review next week, Monday sometime, if possible, in here. And usually, exam 1 is pretty straightforward. I won't say it's simple, because nothing you should take it for granted. And you should get as good as you can. Honestly, for the lab section, final exam is usually not doing very well, because in the final exam, we have 3 labs talk about biochemistry testing. And that is always difficult a little bit. So lab midterm exam, you should do very well in order to help you to make up some questions, so to make up some points. Therefore, I have an insurance policy, which is your lab notebook, 20 points. That's the reason, because final exam, we have a 3 to 4 lab section talk about biochemistry. That always struggling people. Okay? So, not surprised to me, you should do very well in the lab midterm exam. We also will have a little bit extra points for you. So, we wrote it about like 50 points. We maybe make up like 55, the total. So you can make up some here and there. Okay? We expected that most of you could get at least 45 and the NOS. So, which means 45 plus. Okay? So that's all I have for today.